Okay, so the third recording of lymphatic immunity is going to address the third line of defense, which is adaptive immunity. Specifically, we're going to start with cell-mediated immunity. So cell-mediated immunity, this is the first arm of the adaptive immune system. This involves different classes of T cells, which include your CD4 cells, which are your helper T cells, and the cytotoxic T cells, which are your CD8 cells. Note that the CD stands for cluster of differentiation. These cells respond primarily to cells infected with intracellular pathogens. So these are actually responding to your cells that have been compromised. This would include viruses, intracellular bacteria, as well as cancer cells and foreign cells such as those from a transplanted organ. T cells are formed in the bone marrow. They leave the bone marrow to migrate to the thymus to mature. T cells undergo gene rearrangements that lead to a huge variety of genetically distinct T cells. So the genes that actually are involved in making these receptors, there's not many of them. It's just that you end up with the introns and exon rearrangements that give you many, many variations. Each population of T cells that can respond to a specific antigen is known as a clone. There are millions and millions of different clones in the immune system, but only a few cells of each clone exist in the body at any given time. Some T cell clones are capable of recognizing and responding to pathogens, whereas others are not. The thymus actually screens these cells and mediates the destruction of those clones that cannot recognize antigens. The thymus ensures that the individual is immunocompetent, which means that it is able to mount a normal response to a foreign substance. So you have cells of the lymphoid line will divide in the bone marrow. The immature T cells will exit the bone marrow and migrate to the thymus. Once in the thymus, they will go through the maturation process, and the destruction of any self-reactive T cells will occur. And then now the mature T cells will then go and live in some type of lymphoid tissue. Other T cell clones known as self-reactive T cells. These will react to your own cells as foreign substances and would attack your cells if released into circulation. Self-reactive T cells must be destroyed, ensuring self-tolerance and preventing T cells from attacking the self cells. T cells that survive the thymus screening are released into circulation when they mature, and they are referred to as naive T cells, because they have not yet encountered their specific antigens. So let's define what an antigen is. An antigen is a substance that either a B cell or T cell will recognize. The name antigen comes from the phrase antibody generating, in reference to their ability to generate the production of antibodies from the B cells. It's usually peptides, but it may also be substances such as carbohydrates, lipids, and metals such as nickel, as well as other substances, must be coupled with some type of peptide in order to be fully recognized as an antigen. Only certain antigens are called immunogens. These are capable of generating a response from the immune system. Antigens present on your own cells are referred to as self-antigens. And these self-antigens are not immunogens in your body. They can't be. Haptens, on the other hand, are very small antigens. They're immunogenic only if attached to protein carriers. And an important example is urushiol, which is a toxin in poison ivy. This enters the skin, is oxidized, and the product binds to skin proteins. The T cell receptors are found on the surface of every T cell. The receptor must bind a specific antigen. before the cell can become activated. A unique portion of antigen to which the receptor binds is known as an antigenic determinant. And actually, when they're looking at making vaccines, what they try to do is just make the components referred to as antigenic components instead of a whole cell, live cell, or you know, uh, using a whole cell or a live cell as a vaccine. They can just use these little bits referred to as antigenic determinants. The T cells can only interact with pieces of antigen bound to glycoproteins called major histocompatibility complex molecules. The name comes from the fact that MHC molecules are major determinants of compatibility among tissues and organ donors and recipients. The MHC molecules are found on nearly all nucleated cells except the erythrocytes, which make blood products considerably more easy to donate than other organs in the tissues. MHC molecules will serve as docking sites for specific components of antigens that are then displayed as T cells. There are two types of MHC molecules: class 1 MHC molecules, which are found on the plasma membrane surface of nearly all nucleated cells. And then class 2 MHC molecules, these are only on the surface of antigen-presenting cells. Helper T cells generally interact with class 2 major MHC molecules, and class 2 MHC molecules are present present exogenous antigens which have been taken up by these antigen-presenting cells, and they present them to, say, a helper T cell. Exogenous antigens originate outside the cell. They must be taken into the cell by phagocytosis. The endogenous antigens are those that originate inside the cell. Foreign antigen is present on the pathogen that lives inside your cell, and that's what we refer to as endogenous antigen. The exogenous would be a bacterial that was ingested and then now is being presented to, say, a helper T cell. Examples of foreign or self-antigen are encoded by your DNA. You have normal self-antigens. Foreign cancer antigens are made by mutated DNA. Foreign viral antigens, when a virus infects a cell, it inserts its genetic material into the DNA and triggers the synthesis of its proteins, including its own antigens. Endogenous antigens can be foreign or self, but exogenous antigens are always foreign. Basic steps by which the class 1 MHC molecules process and display endogenous antigens. The cell will synthesize either self-antigen or a foreign antigen. Note that some foreign endogenous antigens are not synthesized by the cell. Those are the ones that are from intracellular bacteria. The antigen is broken down by enzymes in the cytosol. Antigen fragments containing antigenic determinants is transported into the rough ER, coupled with class 1 MHC molecule in the rough ER membrane. The MHC antigen complex will then leave the rough ER by a vesicle inserted into the cell's plasma membrane. And that is how the endogenous antigens are going to sort of express the antigen as well as the MHC. Now, the steps involved in exogenous antigens that are displayed by class 2 MHC molecules: the cell will phagocytize the pathogen. The phagocytic vesicle will fuse with the lysosome. The pathogen will be degraded and its antigens are fragmented. The lysosome fuses with the vesicle from the rough ER that contains the class 2 MHC molecules. The antigen fragment will then bind to the MHC molecule, and the MHC antigen complex is inserted into the cell's plasma membrane. So why do we need both class 1 and class 2 MHC molecules? The answer lies in differing functions of helper T cells and cytotoxic T cells. Helper T cells are activated by antigen-presenting cells, bearing portions of the antigens on their class 2 MHC molecules as shown here. Antigen-presenting cells not disease themselves, but rather they function as a warning system and alerts the helper T cells to problems somewhere in the body. The helper T cell then stimulates other parts of the innate and adaptive defenses to combat that threat. Compare this to what happens with the cytotoxic T cell. The cytotoxic T cell is activated by a disease cell that is actually bearing an antigen on its class 1 MHC molecule. When this occurs, the activated cytotoxic T cell lyses and kills the disease cell, which is now its target. The cytotoxic T cells were also able to bind and recognize the class 2 MHC molecule. They wound up killing antigen-presenting cells, not disease cells, would be detrimental to the immune function as a whole. So that's why you have this distinguish. The antigen-presenting cells are presenting exogenous antigens, whereas the MHC class 2 are presenting endogenous antigens, and that whole cell will actually be killed. The process of naive T cell activation is very similar for helper T cells and cytotoxic T cells, and the main difference being the type of MHC molecule that's involved. Dendritic cells have unique ability to undergo cross-presentation. The ability to be able to display some of the ingested viral and bacterial antigens on both class 1 and class 2 MHC molecules, which enables them to interact with and activate naive cytotoxic cells, as well as the naive helper T cells. Both the cytotoxic and helper T cells will activate, the activation consists of several steps, which we'll now go over. So the first step is the dendritic cell displays the antigen fragment on their MHC molecule. The MHC antigen complex binds to the receptor of the specific naive helper T cell or cyto T cell clone. The dendritic cell processes and displays antigen fragments on its MHC molecules. They then present these antigen to a particular T cell clone, specific for its individual MHC antigen complex. The T cell receptor recognizes and binds this complex. Multiple changes triggered inside the T cell activation. This is known as clonal selection because it selects for a particular T cell clone. The second stage is the helper T cell or the cytotoxic T cell binds co-stimulator and becomes activated. The T cell receptors normally have low affinity for their MHC antigen complexes, protective mechanism that prevents unnecessary naive T cell activation. Particularly true is of the cytotoxic T cells because their effects are so destructive when activated. The full T cell activation requires the interaction of naive T cells with other molecules on cells, which are called co-stimulators. The third stage is the activated helper T cell or cytotoxic T cell clone now proliferates and differentiates into effector cells and memory cells. These helper T cells or cytotoxic T cells increase in numbers when activated. They differentiate into effector cells, which cause immediate effects, and memory T cells. The memory T cells are responsible for cell-mediated immunological memory. They respond more quickly and efficiently to subsequent exposures of the antigen. The first time you're exposed to a particular antigen, it takes quite a while. Secondary and subsequent exposures, those occur more frequently. With your memory T cells, there is no need of co-stimulators. And this actually makes for a much, much more rapid response in the future. So this just kind of goes through those three stages again, and you guys can look through these. Just make sure you know the three effects of the three stages of T cell activation. Helper T cell and cytotoxic T cells have very different roles, although they do interact and depend on one another to function properly. The role of the helper T cells is to have no phagocytic or cytotoxic abilities. Helper T cells exert their effects through the secretion of the cytokines that activate and enhance various components of the immune response. In this way, they help the immune response, hence their common name. And it's actually these helper T cells that are required for the normal function of all components of the immune system, including innate antibody-mediated and cell-mediated immunity. Main functions of the helper T cells include innate immunity with the stimulation of the macrophages, with the helper T cells, if these cell lines actually become non-functional, that poses a huge problem for your overall immunity. Essentially, if you do not have functional helper T cells, you are not going to have a functional immune system. HIV targets helper T cells, and this is why people that have very high viral loads and low helper T cell loads, they eventually will enter into what we refer to as AIDS, which they have no effective immune system anymore. Now, let's talk about organ and tissue transplantation and rejection. There are 4 kinds of tissue and organ transplants or grafts. Autografts involve tissues transplanted from one site to another from the same individual. Isografts, these involve organ and tissues transplanted between 2 genetically identical individuals. This would be if you have an identical twin lying around. Like autograft, these result in no response from the cytotoxic T cells because antigens bound to the class I MHC molecules are not recognized as foreign. Allografts, this is the most common type of graft. This involves organ and tissues transplanted by 2 non-identical individuals of the same species. And then finally, the 4th type is xenografts. This is a relatively rare procedure that involves organs and tissues transplanted between 2 individuals of actual different species, such as a pig and human. Both allografts and xenografts contain antigens that organ recipients' immune system recognizes foreign. This leads to a reaction from the immune system that, if left untreated, results in rejection of the organ or the tissue. The rejected organ or tissue first fails to function properly. Its cells die as the cytotoxic cells destroy them. This is a condition known as necrosis. Rejection can lead to death of the transplant recipient from the organ failure and from blood clots and other complications of the necrotic tissue. Graft rejection is something that needs to be prevented, and it's done in 2 ways. The first is ensuring that the allograft antigens are similar as possible to the recipient's antigens. This is determined through a process that screens antigens most likely to cause rejection. Those associated with MHC molecules encoded on genes called HLA genes. The second way that we can help to prevent rejection is suppressing the immune response with medications. These are known as immunosuppressive drugs. Now, transplant patients most likely will remain on immunosuppressive drugs for their entire life. However, what they try to do is immediately following surgery, the transplant drugs are at pretty high levels, and they do try to titrate these back down as time progresses. One of the most commonly performed transplant procedures is a bone marrow transplant, also referred to as the hematopoietic stem cell transplant. And that is something where they will eradicate the entire bone marrow and then sort of transplant that in. Again, you still have to have certain levels of recognition there. You can't just take any bone marrow. Okay, and that is going to be the end of recording number 3. We will have one more recording, which will be the second arm of the adaptive immune system, which is antibody-mediated immunity.